Hello everyone, so today we're going to talk about the cell membrane and its structure and function. And most of us know what the cell membrane does. It's one of the earliest things that we're taught in a general biology classroom. It controls what enters and leaves the cell. But for an advanced class, and really what you should be thinking at this point is, how does the cell membrane know what's supposed to enter and leave? And like most things we're going to talk about in AP Bio, it all comes down to, like as we said, the structure and the function. The structure of something determines its function. So its function involves certain things that enter and leave, so its structure must determine that. So what about its structure makes it so unique, and how does its function actually work? And that's the things we're going to explore here today on Big Orange Biology. Class is in session. Let's begin. So the cell membrane is described at best as a fluid mosaic model. It means it is a mosaic like you would see in an art museum where little pieces are put together to make a larger picture. And it's a fluid mosaic because those pieces that are lipids, proteins, and carbs float around and they bob around in the membrane. They go to different places. So phospholipids form a continuous bilayer, which is like a lake in this case, and allows for a variety of proteins just to float along. And these proteins will move around, and they can be anchored like a boat, but they do move around. They are fluid. Now, what makes the membrane different is that while it is made of phospholipids, it is made of uniqueness. So membrane fluidity is going to be determined because of these lipids by the type of lipids. So for instance, phospholipids that are short and unsaturated are more fluid than, say, long saturated lipids, which makes sense. The unsaturated have the extra double bonds. The double bonds allows for extra movement and posability. So these fatty acids make it a little bit more fluid. Also temperature. Typically, the colder it is, the less fluid the cell membrane becomes. Think about it, because the atoms are moving a lot slower, that they tend to just stick around a little bit. Now, things like cholesterol can be added into the membrane to help regulate this and help maintain certain levels. But as a whole, on the basic level, it all comes down to the type of lipid and the temperature is going to determine how flexible this membrane really is. Now, membranes also contain proteins. Now, these proteins make up a large part of the membrane in addition to the phospholipid. Now, the ratio of proteins to phospholipid does vary, depends on the cell and the cell's function. So, first off, we got the peripheral membrane proteins. These are the ones that are found on the outside. They're on the periphery. Think of like the side of your eyes. Those are your peripherals. You can look out one side and the other, and those are where these same things are going to be, where we're going to find these proteins. So proteins are going to be found on the outside or the inside of the membrane. Now, the integral membrane proteins are going to be partially embedded in that phospholipid bilayer, and that's going to mean some unique functions. So if integral can go inside of the membrane, that means there's something going on special. See, the membrane itself is, hydrophobic tails along with hydrophilic heads. So these hydrophilic heads are what we call polar, and these hydrophobic tails are nonpolar. So if a protein is peripheral, it just needs to be a polar protein. That's pretty okay. It's not impossible to do. Proteins themselves, on a whole, are polar. But for a protein to cross through here and not be rejected and be separated like oil and water, you're going to excuse the bell there, not to be separated like oil and water, it needs to be nonpolar as well. So in order to be an integral protein, that means that part of this protein region, these R groups right here in this little region, have to be nonpolar. If they weren't nonpolar, this would not take place. This protein would be pushed out of the membrane. Now, proteins can be anchored because they also have hydrophobic lipid components that anchor them to the bilayer. Now, proteins are asymmetrically distributed on the inside and the outer membrane surfaces, so they're not in equal amounts. Some proteins are more abundant on the outside of certain cells and more abundant on the inside of other cells. Now, transmembrane proteins are like these integral proteins. They go through the bilayer, meaning, like as I said, they have to have inner and outer parts of the membrane that are polar with a region in the middle that has to be nonpolar. Now, some membrane proteins can move within the bilayers, others are restricted, and we've seen this by fusing other cells. They tend to drift when we put cells together. Now, proteins inside of a cell restrict the movement of membrane proteins, and they can also attach the proteins to the cytoskeleton, because certain proteins you may want on a specific part of the cell for a variety of functions. Also, because of proteins, we can also have cells adhering to each other to help form what we would consider a tissue because of the interactions between the cell surface and these carbohydrates and proteins that we typically would call a glycoprotein. Allows the tissues to actually form. Now, membranes are constantly being broken down, transformed, and fusing, and everything else. Though membranes may be similar, there are major differences of the membranes of even a single cell. So every region of a cell may have a different membrane because of different functions. Remember, structure determines function. If you have different structures, including composition, you're going to have different functions. And that's going to allow it to do certain things. Now, the main function of the cell membrane, as we know, is selectable permeability. These are membranes that allow some substances but not others to cross. And typically, processes of transport can be classified into two categories based on the presence of energy. Now, passive transport does not require any metabolic energy. These are basically substances moving down its concentration gradient, going from a high concentration to low concentration. My mouse didn't want to move there. So as it goes from high to low, that requires no energy. It's like going downstairs, going down a stream, not much effort. That works great. Active transport requires energy. It requires some effort. It's like going uphill. That's because you're going to go from low to high. You're going to have to go through socialization, basically. It's like for some people, like me, I don't like to be around large crowds of people. And for me to go into large crowds, that takes some effort and it can be exhausting. It takes the energy. Active transport's like that. It's when particles have to go from low concentrated areas to high concentrated areas. You're going upstream, you're going upstairs, you're fighting the flow. Now, diffusion is the classic example of passive transport. That's when random movement of particles will reach equilibrium, specifically a dynamic equilibrium. Now, net movement is when there's one side getting more so than the other, but net movement from regions of greater concentration to lower concentrations tend to take place. Now, once equilibrium has been established, there is no net movement, but there's still going to be random movement because it is a dynamic equilibrium. If equilibrium was not dynamic, you would be dead because you ain't having constant movement. If you have constant, only things that don't have constant movement are dead things. Now, speed of diffusion does depend on multiple factors, the biggest one being diameter of the molecule. Smaller molecules go faster. They don't have as much mass, meaning they don't have as much inertia. In a little physics there, but basically without that heavy mass and without heavy diameter, you're going to go faster. Now, temperature also matters. Temperature is going to speed up, so that's one way to overcome inertia is to increase the velocity. To increase the velocity of a particle, you make that particle move a little faster with some energy, and energy is great in the form of temperature. Temperature is a measure of how much energy is in the system. And also concentration gradients. Typically, the more dramatic the concentration gradient, the greater the force that's going to drive diffusion. Something with an increasingly high concentration gradient is going to be more so than a moderate concentration gradient. Extremes favor this. They're going to drive this all together. Now, cell cytoplasm is an aqueous solution, so it is going to be a polar solution along with the surrounding environment. Now, diffusion of a solute. Now, the solute is a dissolved particle, and the dissolved particle depends only on its concentration. So, if you're talking of a solute, dissolved particles only care about dissolved particles. So, high concentration of dissolved particles inside the cell will cause the solute to go out. So, under some situations, the solute will leave the cell if there's too much solute on the inside of the cell. And if there's a high concentration of solute outside of the cell, the solute will go inside of the cell to escape the concentration. However, that's not always the case. See, only some molecules do this simple diffusion process. You got your oxygen gas, CO2, and other small non-polar lipid-soluble molecules. If you're somebody taking notes, those are the ones you really want to take note of. These are really the exception. See, polar molecules cannot pass through. Remember the whole thing about being semi-permeable? Well, this is where it's semi. This is what's not allowing. These are the ones that are not allowing through. Polar hydrophilic molecules cannot pass through a membrane with ease. Now, eventually, over time, it can. It takes a lot of work. And not in terms of energy, but it just takes a lot of time for it to actually make it through. They're not soluble with a hydrophobic interior, so things like amino acids, sugars, ions, and water cannot cross easily. Now, osmosis is the diffusion of water, and you're going to go, wait, Mr. Jennings. You just said water can't cross easily. Yes. Water on its own without any help cannot cross. But water does take advantage of these things called aquaporons, which we'll explore here in a second, that are basically designed just for water. So water is going to travel across just fine. It's great like that. Now, ultimately osmosis is depending on the concentration of water, not the solute. But you can remember this way. Typically, water moves down its own concentration gradient. Meaning water goes from high water to low water. This is going to be determined by solute. Higher the solute, lower the water. So high solute, less water. So this comes up the adage that solute sucks. Solute will pull water towards it. So if you have a high amount of solute, water will travel in that direction. It's complicated to think about for more than a minute, but trust me, that's the best way to keep it straight. Now, when comparing these two solutions, you need to keep in mind the terminology. We have hypertonic, isotonic, and hypotonic. Now, hypertonic means there's more solute on the outside of the cell. So there's solute on the outside, that means there's more dissolved particles on the outside. Water is going to leave the cell dramatically. Isotonic, there's equal salt and equal water on the sides, as we see here. So as water leaves in hypertonic, it shrivels up. And isotonic is equal. So there's going to be equal solute inside and out. And it's great for animals. Plants, they tolerate it. Then there's hypotonic. Now hypotonic, there's more solute on the inside of that cell. Or if we want to keep this perspective correct, so there's more solute outside, equal solute outside, less solute outside. Because there's less solute outside, that means there's more solute inside. And if there's more solute inside, water is going to get sucked towards it. Animal cells do not care for this. Plant cells, on the other hand, love this. Plant cell walls have adapted to basically allow for this to occur. And that's called a turgor pressure. See, concentration of solutes in the environment determines the direction of osmosis in animal cells. Now, other organisms, cell walls will help limit the size of the cell. You can only go so far. The cell walls eventually push back up in there. Now, plants take advantage of this and use what's called turgor pressure. Now, turgor pressure helps build up against the cell wall, allowing for the cell to basically get rigid and allow for the plant to hold itself upright. Now, facilitated diffusion is like diffusion, except it's using proteins. Remember those aquaporons I mentioned earlier? That's an example of those. These are channel proteins that allow certain key molecules and ions to pass through. Now, substances can also be bound to carrier proteins to help speed up diffusion as well, but they all both operate on the concentration gradient, meaning no ATP has been exhausted. Now, ion channels allow specifically ions to go through. Many of them are gated either through a ligand, which is a chemical signal, or voltage gated, which is, well, electricity. Some kind of nerve-based stimulus is going to cause that to open. And as seen here, this is a ligand. It's a signaling molecule. It's like, hey, open the door, and it opens the door, and the ions get to travel across. Yay! It's great for the ions. Now, water moves across, like I mentioned, in the aquaporons. It attacks also on some things like sodium, but typically aquaporons are where it travels. It allows water to travel actually much faster that way, and also along its concentration gradient. Now, carrier proteins, like I said, help facilitate diffusion by binding of substances. Glucose transporter carriers are a great example of this. Now, as glucose is taken into the cell, it's going to be quickly broken down, so there's always going to be a concentration gradient that favors glucose uptake. However, if you have too much glucose, the molecule's not going to be able to transport it in. The carrier protein becomes bound up and it's locked up. Now, in conclusion, there are a variety of cellular processes that allow happen here, but today we mainly cover the passive transports. So the cell membrane allows things to travel across passively sometimes and sometimes actively. It's semi-permeable. Only things that can tend to travel across are the non-polar, small, and maybe some polar compounds. Very small things like oxygen gas, CO2, and non-polar tiny particles can actually travel. On the other hand, that means certain things like water has to take advantage of things like transport proteins inside channel proteins and ion channels, and well, the aquaporons specifically for water. Now, diffusion is when the solute can travel across. That's great, but in biology we rarely get to see that. We mainly concern ourselves with osmosis because the solute more often than not is too polar or too large. So we have to go with water. We have to look at water. So remember, solute sucks. Wherever there's more solute, water's going to go there. That's osmosis. Now in the next video, we're going to talk a little bit about active transport and how we actually determine osmosis. Probably a little different order. So we're going to do some stuff with osmosis for a while. Then we'll get into active transport. That said, It's been a pleasure. This is Mr. Jennings. Have a great day.